My lab studies how plants grow and develop. And one of the questions we ask is, how do cells know their location when they're in a developing plant so that they can become the appropriate cell type? On the left here, we see the plant we use, Arabidopsis thaliana, and each shoot up near where the flowers are coming out at the top has a collection of stem cells called a shoot meristem, which is shown in the middle of the slide. Each of these cells has to assess its position and decide whether it will become part of a new flower or remain a stem cell and part of the stem cell population. Now, how do cells become new cell types? It's long been known that cell type results from gene expression patterns, as shown here with GFP reporters in living plants. And what we're seeing here is the expression pattern, which is dynamic, of regulatory genes that turn on gene regulatory networks, which turn on the protein coding genes that create different cell types, including the cells of the flowers, as seen surrounding the shoot meristem. Now, the gene expression pattern is a scalar field. Each gene has some number of expression level attached to it at each place in the field of cells. Cells are presently thought to learn their positions and therefore their gene expression pattern either from their parental cells, which is not the case for our plants, or from morphogens, chemical substances that diffuse or are transported from cell to cell as signaling molecules. In our plants, an example of a morphogen is indole acetic acid, or auxin, a plant hormone. When auxin reaches high concentrations, a new leaf or flower forms. At the top of the slide, we see a reporter for auxin in the meristem at high concentration in positions where, in the future, new flowers will begin to form. Auxin moves from cell to cell via a transporter network. The transporters are in the plasma membrane of the cells, and they send auxin out of each cell directionally, which creates a net flow of auxin through the tissue, which opposes the diffusion of auxin, which is also occurring at the same time. Now, the position of new flowers is determined by the direction of auxin transport. What controls the direction of auxin transport? That is the polarity of the auxin transporter, which determines where the auxin flows and reaches high concentration. Polarity is a vector, not a scalar quantity, so now we have a morphogenetic vector field. The field can be described fully with differential equation models. And then we have to ask the question, what regulates the polarity of the auxin transporter in each cell? It turns out, after much experimentation, that it's mechanical stress in the cell walls that surround each cell. A cell high in auxin expands and stresses the wall with its non-expanding neighbor. This causes the auxin transporter to go to the plasma membrane adjacent to that wall in the neighbor and pump auxin toward the high auxin neighbor. And it's this that creates the dynamic flow, that is, the morphogen vector field. The mechanical forces control the morphogen vector field. The stress field is a tensor field. So our overall morphogenetic field is a tensor field and includes mechanical as well as chemical signaling, a new way to look at development and at morphogens. What we see here on the left is a finite element method model, which shows that if we poke a hole in a meristem, we create a new stress pattern because the epidermis is under tension, and we can treat it as a thin wall pressure vessel. On the right, we've poked a hole in 3 cells, and we can see over a few hours the rearrangement of the auxin transporter to the walls that are experiencing the most stress. What senses the stress? We're now hunting for the stress sensors and for the downstream pathways that result from stress and change the gene regulatory networks in each cell. One stress sensor is calcium, which is activated by mechanical stress in the cells and creates a wave that moves all the way across the meristem. Here we're poking the meristem with a glass rod, and you can see the wave of cytoplasmic calcium concentration increase, moving across the meristem. And without this calcium signal, the auxin transporter will not rearrange to the stress pattern. Our current work therefore uses genetics, genomics, synthetic biology, and advanced image analysis to find the stress sensors and the pathways that lead from them. We also use computational models. One of the calcium wave is shown here. This is leading to a new understanding of pattern formation, in which morphogens are physical as well as chemical, and vector and tensor as well as scalar fields. Our hunt is on now for the mechanical sensors for all of them, and the way that the tensor field couples with the vector field of morphogens, which couples with the scalar field of gene regulatory networks, so as to have a complete description, computational as well as biochemical, of how it is that plant cells find their position in a developing tissue. Here's what our lab looked like before the COVID epidemic when we could all gather together. And here's what it looks like now. Our group meetings are on Zoom. Thank you very much.